Vanakkam. Welcome to this course on classics in neuroscience. In this series of videos, we have been looking at neural circuits, specifically cortical columns and cortical processing. In the last couple of videos, we looked at the work of Jerry Letwin. Then we looked at the work of Richard Jung and Denis Alph-Fassad, who were recording intracellularly from the brain. In this video, we continue our discussion of intracellular recordings from the brain. Specifically, we will discuss the work of Charles Phillips. Charles Phillips was born in 1916 and he came to Oxford in 1935, the year when Sherrington retired. For some time he studied with Jack Eccles before Jack Eccles moved to Australia. Then he served in the war. After the war, he continued his study with Liddell. In his collaboration and in his work with Liddell, he studied the movement of extremities in response to focal shocks that was given to motor cortex. So, Charles Phillips is recognized for his work on intracellular recordings from the cortex, one of the first intracellular recordings from the cortex. In his collaboration with Liddell, he found that due to focal shocks, these focal shocks stimulated populations of pyramidal neurons that went on to activate specific muscles. These groups or populations of pyramidal neurons he called as congeries of neurons. So, he tackled the difficult problem of intracellular recordings from the cat in 1956 and from the baboon in 1959. In these studies in cats and in baboons, he tested the motor neuron theory and he found that in the largest cells, that is called as the Betz cells, he found that properties were similar in terms of action potential generation, synaptic potentials and integration of excitation and inhibition. He also found evidence for a relatively new phenomenon called as recurrent excitation and of course lateral inhibition. Recurrent inhibition was already shown in the spinal cord circuits, for example, the Renshaw cell circuits. But recurrent excitation is something that is unique to the cortex. This also means that recurrent inhibition as a organizing principle is present or is found in multiple levels in the central nervous system, both in the spinal cord and also in the brain and higher levels, even including the cortex. These studies by Charles Phillips essentially laid the foundation for future studies of both excitatory and inhibitory local circuits within the brain. With this, we come to the end of this video. In this video, we continued our discussion of intracellular recordings from the brain. Specifically, we looked at the work of Charles Phillips, who demonstrated the presence of both recurrent excitation and lateral inhibition, showing that lateral inhibition or recurrent inhibition is a concept, is a phenomenon that is found not just in spinal cord but also in the brain, specifically in the cortex. But recurrent excitation is something that is found only in the brain. Thank you for your attention.